talk about molecular biophysics, and I'm currently at this place, fantastic place, BITS-Pilani, Hyderabad. They're also like SRM that Professor Ajay Ghosh talked about, are building up big in science and deep tech, and that's why I'm there. And till very recently, most of the work that we'll hear about is done while I was at TIFR Mumbai. And what we'll talk about is one sliver of the stuff that we do, but I will try to connect it to the requirement, I would say, of experimental scientists to build their own instruments. And I think I can't stress that enough. And as I go along, I'll try to point out where these things were required. Before I start with that sliver, which will be on membrane, let me quickly run you through some of the stuff that we do, because many of people here won't know me. So this is a great opportunity for me. Protein aggregation, which trying to find out the molecular mechanism of neurodegeneration, such as Alzheimer's, Parkinson's, or non-brain things such as type 2 diabetes. They all happen because the causative agent is supposed to be protein aggregates, not viruses, not bacteria, protein aggregates, your own proteins aggregate. And this molecular mechanism has been fascinating to find out. And it's still, of course, a work in progress. And throughout many years, I'll concentrate only more last 3-4 years, but for many years we have been working on it, where we came, okay, this paper is now published. Where we came to is we looked at how they start to aggregate when they first form the small aggregates, oligomers. And we always thought, because there's a missing piece of the puzzle every time we went in, either in vitro or in cells, that it cannot be just this one thing, because nobody can really reproduce in the test tube what are the conditions are there in the brain. It doesn't, even the logic doesn't apply. The concentrations that you require for aggregation to happen in the test tube are orders of magnitude higher than what is there in the brain. So I always thought there is a partner or partners in the brain that even biophysically, if I put that back in the test tube, will show the same thing. So finally, last year we got to that. Sorry, the reference is missing, but this came through this ApoE being the partner. Other thing that we have been doing for a long time is building microscopes to look at how the brain functions, especially parts of the brain or brain cells which give you feelings of reward, and it gives you a high when you're doing drugs. I mean, I'm not saying you, in general, when you're doing drugs. And we've always wanted to find out why that is happening, how that is happening, what are the other changes that are happening in the neurons, and nobody had a way to look at the main molecules, the neurotransmitter molecules here, 2 of them are there, serotonin and dopamine, how they go about in a living cell. There was no way of seeing it. So that we developed over the last few decades. And this is one of the, used to be the mainstay of my lab. Developing microscope still is, and I'm developing a new type of microscope now, no longer looking at the brain, but looking at cancer tissue that I won't get a chance to talk about. But these sort of made the background that one can build a UV microscope to look at cancer tissue very fast, hopefully intraoperative, and something that has not been done anywhere in the world, but hopefully we are close. The stuff that we'll talk about is membranes, so I would come back to it and not spend time here. The other thing that we talk about, and sometimes talk to Raghavan quite a bit about this, is looking at peptides and self-peptides. Take a piece of a protein, present it to itself, of course, stabilized in a conformation that it looks like it does in the protein. So if you take a piece of the protein, it no longer looks like it because it sort of unfolds. So you keep it folded chemically, and then wonderful things happen because this protein recognizes this self-peptide, and we're having a lot of fun with it, making protein silencers with this self-peptide, and probably have a chance to hopefully, at least in a laboratory, go to antibiotics. This is the other thing that I said is developing technology. Many of the research that we have done, there's always been an element. I've been trained as a physicist, doing some biology, meddling in some biology, and used to head the chemistry department in TIFR. That's sort of as interdisciplinary as you can get. But heart has been my training as a laser spectroscopist, understanding optics and laser spectroscopy and building tools. That's been a passion, and we have done it over the years. We have taken, I think, as I said, I believe that we experimentalists in India, we need to start making our own tools. As many of you do, heading some department or something with applications, people are saying, okay, I need an instrument that costs 4 crores, 5 crores, 10 crores. I go back and ask, can you build at least part of it? Do you understand it well enough? And people who are very good scientists, they publish very well, unfortunately do not understand their instruments. And this cannot go on in 2025, given the geopolitical situation and the economic situation. We must start building our own instruments. We've started doing that in a little way. Not the ones that are saying that, hats off to those which says, okay, much cheaper, etc. But I'm talking about things which the world doesn't look at because it's cheaper, because it's better than what can be done. So some of these instruments, fledgling efforts, but now I am going in a much bigger way. And I also started teaching people students who would become tomorrow's scientists, knowing how to build these things, break the instrument apart, put it back. And this has been going on now, the 16th year now, something called FCS, started by teaching people how to build this instrument, but now it's much bigger, all of fluorescence and Raman microscopy. Now, lots of my friends from all over the world and in India run this. And, about, 1,500 graduate students have gone through this. Many of them have done wonderfully in building methods, instruments. Unfortunately, not so much in India. They have all gone abroad, etc. But this is something that is very close to my heart, and we keep doing it. Okay, so come back to today's talk and talk about membranes. So for those who are not biologists, I understand, and there are some, I met a doctor on the way here, some were on this side of the spectrum, some are on that side of the spectrum. The cell has membrane, which is a barrier that separates it from the outside to the inside, okay? And it also has internal membranes, which are barriers which allow it to do different chemistry in different locations inside the cell. And that chemical organization, ultimately, what makes life. So it's very important as a barrier. So you can think of it as a wall. But if I tell you that you're building a wall, then you'll say, okay, give me some regular sized bricks and I'll build you a nice wall, because they need to fit with each other properly. And I tell you that nature has, and if you'll pick the right kind of brick, but you will not mix and match them, because it's going to be a very tough thing building a proper wall if you mix and match them. But the bricks that nature uses for building these walls, the membranes, are of 200 different kinds. And I dare say nobody really understands why we have so many types of bricks here, the chemical bricks, let's say the lipid molecules, cholesterol being one of them, and other things. Why we have so many? I mean, it's very difficult to make some arrangement if you just throw in randomly, quote unquote randomly, many different things. And this has really been my passion to try to understand this over the last 4 years or so. And the answer, of course, that it's just not just a brick, you're not making this barrier just to make a very solid barrier, because the barrier that life has between the organelle and the cytosol, or between the cell and outside, has to do many more things than just making a brick wall. So this is a molecular level cartoon of a membrane. And you see there are different types of lipid molecules. There is a lot of disorder in this. There are other things. Other than the 200 types of lipid molecules, many types of proteins, membrane proteins, and stuff that we have started focusing on the last 4 years, many small amphiphilic molecules. You just heard about amphiphilic molecules from Professor Ajay Ghosh. These are biological, natural amphiphilic molecules, and people have neglected the fact that they can go in at very high concentrations into the membrane and perhaps change its properties. Of course, there have been many Nobel Prizes on these things, which are proteins which are there in the membrane, including how they allow the membrane to do some of its function. Thomas Sudhoff got his Nobel Prize looking at endo and exocytosis, how some proteins in the membrane and other proteins actually help that process. So it has to do, so the membrane is not a brick wall because it has to do many things, including something that we'll focus on is endo and exocytosis. Endocytosis is the membrane invaginates in and takes things inside the cell. Exocytosis is there are already preformed vesicles, balls of chemicals which go out, and this is thrown out to the outside world. We need to talk, we need to communicate, then that makes us human, and the cells need to communicate to survive, okay? Both transport and signal communication, transport of material signal communication is very important. And cogito ergo sum, I do exocytosis, therefore I am, because in the neuron, when you are listening to me, this is exo and endocytosis processes are going on all the time. Not only in neuron, it happens in all cells, but signal transduction between neurons happens primarily through chemical means. These molecules which I mentioned, such as serotonin, they're made inside the neuron, they're packaged in a vesicles, they go, which is also a membrane-limited compartment, goes to the outer cell membrane, it has to open it up and throw out that thing when it needs to, quote unquote, talk to the next neuron. So the question that we have been asking is that the membrane composition, many of us take cholesterol-lowering drugs. Cholesterol-lowering drugs because that is inhibiting the synthesis of cholesterol. Question is, what does it do? Because it will probably change the membrane composition in some way. And does it affect other functions of the membrane? We are all happy that its cholesterol is being lowered, but what else can it do? That's a question that I'll give you a little bit of an answer to, a slightly disturbing answer, but anyway. So you see these little, so these are molecules, they're in, the molecules, of course, are not to scale. They're in something called vesicle, and this process, this vesicle, one of these has gone in and has merged here, and this is the process of exocytosis. So we're very interested in knowing how does exocytosis get controlled. As I said, Thomas Sudhoff and others got a Nobel Prize telling us how proteins control exocytosis, how chemical signaling with calcium causes exocytosis. But this story doesn't take in the fact that there has to be, the membrane has to be pliant enough to do it. If I make a very strong big brick wall, it's very hard to make a hole through it. So nature has to make it pliant enough, and whether these small molecules, which are anyway part of this, are playing a part in making it pliant enough, including the neurotransmitter that's inside. Okay? And we started this work about 4 years ago or so, and not too many people are thinking about it. Now quite a few people are thinking about it. So I took my privileges as the executive editor of physical chemistry to commission a special issue, because there are 36 accepted articles already coming up next month. So if anybody is interested, look at that virtual special issue. Okay, so quickly, what questions are we asking? And can small, how much time do I have?
7 minutes.
Huh?
7 minutes.
Oh, 7 minutes, okay. So 4 quick questions. Can small amphiphiles change the properties of lipid bilayers? This is something that has been neglected. How does that affect other things that the membrane has to do? The vesicle association, fusion, and membrane protein dynamics, properties that are different from a brick wall. Is this relevant for living cells? After all, that ultimately what matters. We'll find it out in artificial bilayers and look at it in living cells then. And then finally, can we find or design molecules which are able to do this? Then I have molecules which only talk to the membrane, not to any of the proteins. They're quote-unquote bioorthogonal except for the membrane, but I know exactly how they change the properties of the membrane, so I can control exocytosis, endocytosis. Can we? That's the question. And the example molecule is serotonin, which is a natural amphiphile. So first question, can small amphiphiles change the properties of lipid bilayer? When I say properties of a bilayer, it's a very complex object. Which property am I talking about? And we also didn't know what property to look at. So we targeted about list of 9 properties, and we had to build an instrument which can measure all those 9 properties, or most of those 9 properties together on the same membrane at the same time. So this is where, again, I say that an experimentalist should be tools. There are undue advantages that you get for doing it. There is an AFM, atomic force microscope, confocal microscope, fluorescence lifetime imaging microscope, and fluorescence correlation spectrometer built by my students, not by me. And other techniques such as solid-state NMR, this work has been frequently in collaboration with my very good friend Daniel Huster. So he does solid-state NMR. So this is AFM image, the solid-state NMR, order parameter measurements. Sorry, you don't have time to go into this. But the point is, if you put few millimolar serotonin, and few millimolar is not very large. I have to finish quickly. You have 250 millimolar serotonin packed in your vesicles in your neuron. So it does affect the mechanical properties. It does affect membrane order, does affect how easy it is to make a pore in the membrane. It does affect membrane protein function by changing, not talking to the protein itself, but talking to the membrane and doing some kind of membrane-mediated allostery and changes the functions of GPCRs in the membrane. And it does change the ability of vesicles to fuse. This is sort of a mimic of exocytosis in a test tube or in a cover slip, artificial system. And you see that it changes the rate at which vesicle fusion happens. It may not be only the proteins which control it, okay? And the cautionary tale that I said I will say is that if you reduce cholesterol, somehow the ability of the membrane to make a pore reduces. So your ability to do exocytosis actually reduces. I'm not scaring you, but there is a recent report in Britain looking at 5,000 cases of people who are doing statins for the first year, and the intellectual ability goes down. It recovers over 7 years, which is interesting because body has this thing, so don't be too scared. But we're trying to find out the mechanical origin of this, and this may be something. Is it relevant for living cells? Yes, it is. We looked at serotonin and membrane order in vesicles by again building a very new kind of microscope, and we found that if you look at membrane order, these are neuronal cells in culture, and you see exocytosis after inducing exocytosis. The vesicles are the yellow, they go out, and they become stiffer. G by R is a measure in a spectral microscope of how stiff the vesicles are, and this is with time, it goes down, the stiffness goes down. That is, less stiff vesicles are getting released more preferably. So you need to be pliant enough to be released. Last question is, can you find molecules which are able to do this effectively without talking to others? We started with something which is a psychedelic. Okay, there is something that needs a license for large quantities and for any quantity in Germany, so Daniel couldn't do it, and we could. Small quantities are available here, and in Kaustubh Mote did some solid-state NMR. We found out that we start with the psychedelic, which is of course not bioorthogonal by any means. Try to understand the chemical nature of why this psychedelic is actually 100 times better than serotonin in perturbing the membrane. It can perturb it at micromolar level. So we went through many molecules, looking at these 9 parameters and a set of parameters, trying to measure where they go in, how much they do, what they do, why they do it. And we have some sort of quasi-bioorthogonal molecule, NAS, N-acetyl serotonin, this which seems to be a starting point for us to think about how to make a molecule which can do many things, make the membrane plastic without talking to any of the receptors. So this is what I told you. Can small amplifiers change the properties of lipid bilayers? They can. All these can be, so can these affect function of bilayers such as vesicle association, fusion, etc., important for exocytosis, endocytosis, that they can, and this is relevant for living cells. I just showed you some cell pictures. And can you find the chemical logic of why some molecules do it and some molecules don't, or they do it in orders of magnitude different efficacy? And I think the chemical logic is emerging, so that new class of drug molecules which work solely by membrane modulation is in principle possible. My last slide is that we had to build a lot of instruments, and this is hard to do in India. Over the last 2.5 decades that I've been here, it's extremely hard because the little screw that you need will take 3 months to come. But we still need to do it. We still need to encourage people, young people who are coming in to do it, because there's no longer time that you can just ask for a 5 Pro instrument and produce some papers with it. We better make our own instruments, as whoever can. And you look at China, every time I go to China, it's just mind-blowing by what leaps and bounds they're progressing. So let me just thank my students, my collaborators. I don't have time to name them, but the three ones are the Ankur Gupta, Deb Shankar, and Ankur Chaudhary, two Ankurs and Deb Shankar, who did this membrane work. Funding has come from different places, and my collaborators are there. And I'll end it with things that I would not talk about, but I exhort you to think about this. Technology, and build a community. I've just started doing it in BITS-Pilani, and hopefully you'll give a platform like I did by starting the conference. An industrial platform, you come in with your ideas, go out with a prototype. If it's something optical, do talk to me. Thank you very much.
Thank you, Sudipta. Fantastic talk, inspiring as always. We've got time for a couple of questions.
So a totally technical question. You said G by R is some measure of stiffness. It's a purely optical, microscopy-based measure. How does that, what do you do?
Okay, sorry, I didn't have time. G is green, R is red. It's a spectral shift of a reporter molecule that goes into the membrane, and it becomes stiffer when it becomes greener. So
I see.
The spectral shift, and you can measure it in the microscope while you are measuring serotonin concentration, etc. that we built, but G by R, and we can look at individual vesicles going out. So we can say, okay, that guy has that vesicle's name is Shriram, and that has a G by R of this. Its probability of exocytosis, is it large or not? That is clearly correlated with the G by R thing, which also is correlated with how much serotonin it has in the membrane.
Nice. Sorry, is DOI a well-known psychedelic? Does it have a market name?
Okay, so DOI itself is not so good. There are things, have you heard of magic mushroom? So yeah, so magic mushroom or psilocybin is in that family, and DOI is, it probably doesn't have too much market value because you have better things.
So I want to know, you mentioned that the concentration of serotonin itself affects the exocytosis phenomenon. So what is the mechanism? I mean, and do you think it's true for the other neurotransmitters as well?
Very good question, thank you. I think it's true for the monoamines. And I think there is a very good reason. Of course, I'm just guessing here, which is the monoamines have a tendency to go into they're very amphiphilic. They concentrate in the membrane, and they are known to have after effects. They are neuromodulators, and they are not just point-to-point, millisecond to millisecond communication thing. When neuromodulation, serotonin, for example, is in the pathway of depression, and what Prozac does, which is an antidepressant, is actually increases the population of serotonin in the extracellular space where it can go and sit on the membrane. It partitions into the membrane phase about 1,000 to 1. From the thing. So I think, and dopamine to some extent also does similar things. So I think these monoamines have, the neurotransmission due to monoamine has a long-term memory in the system, and that's why it's neuromodulatory, not just neurotransmitter. I don't know if that answered the question, but I'd love to talk to you later about it.
Thank you.
Sure. Shona?
Yeah. Hi, Sudipta. Wonderful talk.
Thank you.
Are you suggesting that the increased ply nature of the vesicles would also increase drug efficacy eventually in terms of delivery?
Yes. So if you modulate the nature of the vesicles, the mechanical properties that you can actually measure, I think it will absolutely also modulate the effects of drugs. Absolutely. Yes. That's exactly what I'm pointing.
Sudipta.
Sorry.
Yes.
Hi.
Pleased to hear about building your own equipment. But in your talk, you're talking about AFM, solid state NMR. Can you just give some more clue about what fraction of it is home built and what is bought?
Okay, you are asking the technical engineering question.
No,
so that
I am all for building your own equipment, but just when you say AFM, was it bought or was it made?
Yeah, so it is AFM, atomic force microscope.
Yeah.
Confocal, which is a type of fluorescence microscopy, fluorescence lifetime imaging, which tells you the fluorescence lifetime, you know about limb, and FCS, which is fluorescence correlation spectroscopy, which tells you about.
All of it was built here, or you bought some of them, and that was the.
What? Sorry.
All of it was built by you, or
all of it means okay. let me characterize that thing. When I say building, suppose I want a PMT or this thing, I don't build a PMT. I
buy
get
a-
it from Hamamatsu.
Sure.
But the value addition and the ability to make an instrument that nobody else has really is in putting the sort of components
Optics.
together, designing the whole optics, making your microscope and putting it to. The AFM head was purchased because I had it from long time ago and I just ripped apart from the existing AFM, put it on. And
Thank
like
you.
this. Yeah.
Heads off.
But there is an AFM builder in India, and he was in Bangalore, Shilp Sciences. And I'm sad to see that he didn't make much headway. But we need to get these people together and have a platform where everybody has like 10% of the ability to make the whole instrument. But if we can have this platform together, come on in, we'll give you a platform, we'll give you other people who can do the other parts. I think we need to do that and start making real instruments which really sell and actually can be marketed.
Yeah, thank you, Sudipta.